I'm here to present on the topic myoglobin from biomolecules. To start with the introduction, myoglobin, the first protein whose structure was determined by X-ray crystallography, which is a small protein with relatively simple oxygen binding behavior. And its X-ray structure was determined by John Kendrus in 1959. This slide shows about the structure of myoglobin. Globin means protein and myo is muscle. The protein which is present in the muscle is said to be myoglobin. And it is the functional unit of myoglobin is an iron porphyrin complex that is embedded in the protein, which contains 153 amino acid residues and that resembles beta subunits of hemoglobin. And the molecule contains 8 regions of a helix implying mobility and a single iron porphyrin group located in a cleft between helix E and F. And heme ion is 5 coordinate with the fifth ligand to the ion is provided by a histidine N from helix F. The sixth position is the site at which oxygen may reversibly bind. So this is the structure of myoglobin, which I have explained in the previous slide. And this slide shows about the oxygen binding to myoglobin. So myoglobin coordinates oxygen reversibly and control its concentration in tissues. Deoxymyoglobin, that is MB, is bluish red and contains Fe2. And this is the oxidation state that binds O2 to give the familiar bright red oxymyoglobin. And this ion in deoxymyoglobin is 5 coordinate, high spin and lies above the plane of the ring. When O2 binds, it is coordinated end on to the iron atom, the electronic structure of which is tuned by the F helix histidine ligand. The unbound end of the oxygen molecule is fastened by a hydrogen bond to the imidacyl group, that is imidacyl NH of the histidine in helix E. And in some instances, deoxymyoglobin becomes oxidized to ion 3, which is called methmyoglobin and is unable to bind to the oxygen. Then spinning states. The coordination of oxygen which causes the ion 2 to switch from high spin to the low spin, where no d electrons in antibonding orbitals to shrink slightly and move into the plane of the ring. And second point will be the bonding is often expressed in terms of ion 2 coordination by singlet oxygen, in which the doubly occupied antibody 27 orbital of oxygen acts as a donor and the empty 2 pi, that is orbital of oxygen, accepts an electron pair from the ion. And an alternative description is recently considered in which the bonding is expressed in terms of low spin ion 3 coordinated by SO2, in the sense that is alpha, beta and 4 subunits. In this slide shows about the oxygen binding curve of a myoglobin and hemoglobin. Hemoglobin is a blue color graph and the myoglobin is the red color flag. That is oxygen measurement, that is percentage oxygen measurement. And this slide shows about the oxygen binding of hemoglobin versus myoglobin. As shown by the graph, this low oxygen pressures, the affinity of deoxyhemoglobin O2 is substantially lower than that of myoglobin, whereas at high O2 pressures, the two proteins have comparably oxygen affinities. The physiological consequence of the unusual sigmoid shape, that is S-shaped oxygen binding curve of hemoglobin or enormous, and in the lungs, where O2 pressure is highest, the high oxygen affinity of deoxyhemoglobin allows it to be completely loaded with O2, giving 4 oxygen molecules per hemoglobin structure. And in the tissues, however, where the oxygen pressure is much lower, the decreased oxygen affinity of hemoglobin allows it to release oxygen, resulting in a net transfer of oxygen to myoglobin. And the shape of the myoglobin's oxygen binding curve is hyperbolic, as I mentioned you earlier, meaning that it holds onto oxygen much tighter. That's what it is said to be hyperbolic. And only when the amount of oxygen is extremely low, as in the mitochondria of working muscle, does myoglobin release its oxygen. There, the oxygen is used in cellular respiration to produce energy. Then this slide shows about the functions of hemoglobin versus myoglobin. The distinct binding curves, that is a graph which is shown in the previous slides, shows that protein reflects their functions. Two proteins, that is hemoglobin and myoglobin, reflects their particular functions, and hemoglobin, which is well suited for oxygen binding in the lungs, transport in the bloodstream, and delivery to the tissues, and myoglobin, which is well suited for oxygen storage in the muscles and delivery to mitochondria whenever it is needed. And myoglobin does not exhibit cooperativity nor Bose effect. And this slide shows about the difference between hemoglobin and myoglobin. So hemoglobin and myoglobin differentiation. In this primary structure, myoglobin is having 153 amino acids, whereas in hemoglobin 141 amino acids, that is alpha chain, and 146 amino acids, that is beta chain. And in secondary structure, that is in myoglobin, it has 8 alpha helix, and hemoglobin 8 alpha helix for each alpha chain and beta chain. And regarding tertiary structure, myoglobin has folding of alpha helix and hemoglobin has folding of alpha helix too. And quaternary structure, myoglobin has none of the structure, and hemoglobin has tetramer of 2 identical alpha beta dimers, that is subunits. And regarding functions, myoglobin has oxygen storage and hemoglobin has oxygen delivery. And what are the disease which causes myoglobin? What are the disease which occurs if this myoglobin doesn't occur properly? It is acute renal failure, heart attack, myoglobinuria, that is rhabdomyolysis. And causes of myoglobinuria, MC-Ardell's disease. So what is this MC-Ardell disease? This MC-Ardell disease is a genetic disorder that mainly affects skeletal muscles, and it occurs due to the deficiency or absence of an enzyme called myophosphorylase, and the symptoms like painful muscle cramps, weakness, and fatigue manifest during periods of physical activity. And second one will be phosphofructokinase deficiency. Next, carnitine palmitoyl transferase 2. Fourth one will be malignant hyperthermia. Fifth one, polymyositis. Sixth one, lactate dehydrogenase deficiency. And seventh one will be thermal or electric burns. And how it is diagnosed. So after centrifuging, the urine of myoglobinuria is clear. That is, the urine sample is being centrifuge. So what it is diagnosed as myoglobinuria is clear in centrifugation process, and serum of myoglobinuria changes from pink to red color. So overall, from this slides, it has been shown that myoglobin facilitates oxygen diffusion, and myoglobin desaturates at the onset of muscle activity, which increases oxygen diffusion gradient from the capillaries to the cytoplasm, and myoglobin has also been shown to have enzymatic functions. So it is a protein that is found in striated muscles, which includes skeletal muscles, that is the muscle attached to the bones and tendons, and heart muscles. Its main function is to supply oxygen to the cells in the muscles, that is myocytes. Here I am raising a question: does myoglobin store oxygen in muscle cells? Yes, it can store oxygen in the muscle cells. How? It is due to the presence of heme moiety. Myoglobin serves as a carrier and store for oxygen in muscle cells of the body. And this myoglobin has more affinity for oxygen as compared to the hemoglobin. As a result, it can acquire oxygen from hemoglobin, hence transferring it from the blood to the muscle tissues. So what kind of protein will be this myoglobin? This myoglobin is a heme protein which is found in skeletal and cardiac muscles, as I told you earlier. So which food is rich in myoglobin? Myoglobin content is higher in beef and lower in poultry, with lamb and pork having intermediate amounts. The age of an animal will also impact the myoglobin content of the muscles, with older animals which is having more myoglobin and darker meat. Then, is myoglobin a blood urine? When muscle is damaged, myoglobin in muscle cells is released into the bloodstream, and the kidney helps to remove myoglobin from the blood into the urine. When the level of the myoglobin is too high, it can damage the kidney. And then next question arises: is myoglobin a kidney? When muscle is damaged, a protein called myoglobin is released into the bloodstream. It is then filtered out of the body by the kidneys, and this myoglobin breaks down into substances that can damage kidney cells. Okay, so which cells produce myoglobin? So myoglobin, a mobile carrier of oxygen, is developed in blood vessels and heart cells in response to increased demand for oxygen during exercise, and transports oxygen from the sarcolemma to the mitochondria of vertebrate heart and red muscle cells. The next question arises: is myoglobin is an iron containing protein? It is a molecular of 18 kilodalton. It resembles hemoglobin but binds one rather than four molecules of oxygen. So its binding characteristic was such that it takes up oxygen from hemoglobin in the blood and releases it for use into the mitochondria, where oxidative reactions occurs. Then what causes the myoglobin to increase? Myositis. So myositis is the name for a group of diseases with inflammations in the skeletal muscles. So this inflammation causes muscle damage as well. Next question arises is: why is too much of myoglobin is bad to health? Kidney filter a blood for myoglobin so that it can be passed out of our body into the urine. But too much myoglobin can overrun the kidney and lead to kidney failure. So on the whole, which is more important, that is myoglobin or hemoglobin? Our body prefers to use hemoglobin rather than myoglobin as the oxygen carrier in the bloodstream. So this is because hemoglobin not only binds oxygen weakly but more importantly binds oxygen comparatively. Thank you.